Hello all, welcome to my YouTube channel. This is Dr. Dheenadhayalan, Assistant Professor, Department of Pharmacy Practice, School of Pharmaceutical Sciences, VISTAS. In this video, we are going to explore about a critical piece of personalized medicine puzzle that often gets overshadowed by our own DNA, which is nothing but pharmacomicrobiomics. So in this video, we will explore how our resident gut microorganisms influence drug metabolism, therapeutic efficacy, and how modern pharmaceutical science is turning these microbes into targeted delivery systems. So are we truly alone? So let's start with the fundamental question. So when you look at yourself, you see a single organism, but biologically, your body harbors nearly 100 trillion microbial cells. These microbes actually outnumber your own human cells. So, the gut is the most densely populated region, acting almost like a vital organ that constantly communicates with your brain, immune system, and metabolic pathways. Remarkably, the genetic material of your gut microbe is 150 times larger than your own human genome. So, the second genome, because of this vast genetic diversity, scientists refer to a gut microbiome as our second genome. While human DNA encodes roughly 23,000 genes, our microbial partners bring over 3 million genes to the table. Together, host and microbe functions as a collective biological unit called holobiont. So this second genome operates as a dedicated microbial metabolic organ carrying out complex chemical reactions that human and liver enzymes simply can't perform. And what is the biological scope? Taking a look at the colonic landscape, it is predominantly inhabited by anaerobic bacteria. The four primary phyla are bacteroidicides, which make up to 30%, actinobacteria at 7%, proteobacteria at 5%, and firmicutes. So these organisms play indispensable roles in regulating nutrient breakdown, synthesizing essential vitamins, and maintaining the structural integrity of intestinal mucosal barrier. So, pharmacomicrobiomics. In traditional pharmacology, often struggles to explain why two patients give identical drug doses, exhibit completely different clinical responses or side effect profiles. So, these pharmacomicrobiomics bridges this gap by studying how gut bacteria interact with the drugs. Microbial enzymes can chemically transform drug molecules, altering their therapeutic efficacy and toxicity before they are even absorbed into systemic circulation. So, what is the balance of health and dysbiosis? A healthy gut maintains high microbial diversity dominated by beneficial commensals. When this dynamic equilibrium is disrupted, the patient enters a state of dysbiosis. Dysbiosis is characterized by a loss of beneficial bacteria, reduced overall diversity, and an overall growth of potential pathogens. Crucially, for clinical practice, dysbiosis is linked to systemic conditions such as inflammatory bowel disease, metabolic syndrome, and type 2 diabetes mellitus. So, you must know about these terms: second genome, dysbiosis, bioengineering, and pharmacomicrobiomics. Let me pause to consolidate four vital concepts of this slide. The second genome, which is the collective genetic material of resident microbes. Second, dysbiosis, which is the microbial imbalance linked to chronic disease. Third one is bioengineering, applying engineering principles to redesign cellular functions. And the fourth one is pharmacomicrobiomics, which is the field of studying how these microbes dictate our response to medications. a pharmacy inside you. So you can think of your gut as a internal chemical factory. While traditional pharmacotherapy focuses exclusively on treating the host, the patient, next generation medicine takes a paradigm shifting approach by treating or modifying the guest, the microbiome. We can effectively cure or manage disease in the host. Microbes and drug metabolism. So, when an oral drug is ingested, it encounters gut bacteria long before reaching systemic blood vessels. Microbial metabolism typically follows two primary biotransformation pathways. First is activation, where bacterial enzymes cleave inactive products into active therapeutic agents, which is a classic example being sulfasalazine. Second is inactivation, where bacteria degrade an active drug into an inactive metabolite, significantly lowering drug bioavailability, as seen in digoxin. So, metabolic powerhouses. Beyond drugs, these microbes transform endogenous compounds to maintain physiological hemostasis. For example, gut bacteria dehydroxylate primary bile acids into signaling secondary bile acids. They also metabolize dietary tryptophan into indoles, such as indole 3 propanoic acid, which is a potent antioxidant that reinforces barrier immunity. And the living pharmacy. Instead of delivering a static chemical pill, imagine prescribing a living factory. Through synthetic biology, researchers are engineering probiotic strains like E. coli Nissel 1917, which circular plasmid DNA. These smart bacteria sense local intestine inflammation and synthesize therapeutic proteins right at the site of disease. How wonder, which drastically reducing systemic toxicity. The antibiotic paradox. So this brings us to the antibiotic paradox. Antibiotics saves lives, but they function as broad spectrum molecular bulldozers. They wipe out essential commensal bacteria alongside pathogens, triggering acute dysbiosis. So this leaves the gut environment vulnerable to opportunistic infections like Clostridioides difficile. So know about these terms: microbiota, common cells, probiotic, hemostasis. Microbiota refers to specific microorganisms residing at an environment. Common cells are microbes living peacefully within us without causing harm. Probiotics are live microorganisms that confer a healthy benefit when administered. And hemostasis is a steady physiological state maintained by these mutualistic interactions. So, when microbes turn toxic, so there is also a darker side to microbial drug metabolism. Sometimes bacterial action generates toxic byproducts that drive severe adverse drug reactions. A prime clinical example is the chemotherapy agent irinotecan. After the liver inactivates irinotecan, bacteria, bacterial beta glucuronidase enzymes in the colon cleave the inactive conjugate, reactivating the toxic drug into the gut lumen and causing severe intestinal mucosal injury and diarrhea. So, drugs at the microbial interface. So, let's examine three high yield drug microbe interactions. Metformin positively alters the microbiome to assist glycemic control. On the other hand, digoxin is inactivated by specific cardiac glycoside reductase enzymes in Egerbilla lenta, reducing its cardiac efficacy. And in Parkinson's therapy, microbes metabolize levodopa peripherally before it can reach the central nervous system, forcing clinicians to raise doses and accept higher peripheral side effects. So how can we personalize the treatment in this? Because diet, genetics, and environment vary greatly, especially across diverse populations in India. Gut microbiome profiles are uniquely individual. Personalizing medicine requires integrating host genetic markers, lifestyle factors, and gut metabolomics. Given India's high burden of type 2 diabetes and IBD, engineered live therapeutics represent a transformative frontier. So, what is the future in immuno-oncology? So, in oncology, the gut microbiome determines whether modern immunotherapies will succeed. Studies show that cancer patients with high baseline levels of Akkermansia, mucinophilia respond significantly better to immune checkpoint inhibitors. Gut microbes effectively prime T cells to recognize and attack tumor cells more aggressively. So that's why we engineer engineering the smart bacteria. So how do we engineer these smart bacteria? Synthetic biology installs genetic circuits containing a sensor protein and a promoter and a therapeutic gene. So the process follows three distinct steps. First one is detection, where the bacterium senses a specific disease biomarker. Second one is response, where the genetic switches trigger drug expression. And the third one is delivery, releasing the drug locally at the target tissue. So fecal microbiota transplant. When dysbiosis is severe, fecal microbiota transplant offers a total ecosystem reset. Healthy donor stool is rigorously screened for pathogens, filtered and administered via capsule or colonoscopy. So these introduced donor microbes engraft within the host gut, physically outcompeting and displacing pathogenic strains like Clostridium difficile. So this FMT beyond infection. While FMT is currently the clinical gold standard for recurrent Clostridium difficile infections, clinical research is rapidly expanding into metabolic syndrome and IBD. Early trials demonstrate that resetting the gut microbial community can significantly enhance host insulin sensitivity and reduce systemic inflammation. So again, know about these terms: postbiotics, stratification, and biomarker. So let's review these three terms. Defining precision microbiome therapeutics. Postbiotics are non-viable bacterial products or metabolic byproducts that can confer clinical benefits. Stratification is the practice of categorizing patients into subgroups based on their microbiome profile to predict drug response. And a biomarker is a measurable biological indicator in stool or blood used to track disease or therapeutic response. So the next one is live biotherapeutic products. What are these? So it is essential to distinguish commercial probiotics from live biotherapeutics products or LBPs. Probiotics are sold primarily as dietary supplements. LBPs, on the other hand, are rigorously tested, highly regulated pharmaceutical products containing live organisms designed to treat, cure, or prevent specific human diseases. So, precision nutrition workflow. The clinical workflow for personalized microbiome medicine follows four distinct steps: sampling tool and blood, high throughput metagenomic sequencing, AI driven data analysis to identify biomarkers, and precise therapeutic intervention via tailored diet or pharmabiotics. So, as Dr. Iran Ilivinov noted, the microbiome is a dynamic organ that can be systemically reconfigured. So, personalization in action. Here are two concrete case studies. In type 2 diabetes, microbiome profiling explains why identical food cause vastly different postprandial glucose spikes in different individuals. In cancer immunotherapy, researchers are using FMT for treatment responders to reprogram the immune systems of non-responders, converting them into successful treatment candidates. So, what are the roadblocks for these innovations? Despite immense promise, major translational hurdles remain. First, distinguishing causality from mere correlation. Is the microbe causing the disease or just living in an altered environment? Secondly, stool samples do not always reflect mucosal microbes adhering to the gut wall. Third, the results from Western cohorts often fail to reproduce in Indian populations due to dietary variations. So, what are the ethics and regulations? Engineering live organisms introduces unique regulatory and ethical requirements. Data governance must safeguard the privacy of patient's unique microbial fingerprint from a biosecurity perspective. Genetically engineered strains must incorporate genetic kill switches to guarantee they cannot survive if shed into external environment. In India, engineered bacterial therapies are classified as genetically modified organisms requiring strict regulatory approval from the Genetic Engineering Appraisal Committee. As pharmacy professionals, we must also address socioeconomic equity to ensure these high-tech biotherapeutics remain affordable for rural and undeserved populations. So, to summarize our keynotes, the gut microbiome functions as a second genome and metabolic organ. Microbial enzymes directly alter drug efficacy, toxicity, and pharmacokinetics. Synthetic biology enables us to turn common cell bacteria into smart drug delivery vehicles. Translating these therapies into practice requires rigorous regulatory oversight, biosecurity controls, and personalized profiling. So, if we are offered a living pill tailored to microbiome to prevent chronic disease, what would your biggest concern be? Patients often raise questions regarding long-term GMO safety, data privacy, religious considerations, and drug cost. As pharmacists, communicating these biological mechanisms clearly to patients will be vital to building clinical trust. We are rapidly moving away from one size fits all medicine toward an era where therapeutics are tailored to your internal ecosystem. India's biotechnology sector is playing a pivotal role in driving this revolution. Remember, modern healthcare is no longer just about treating the host cells, it is about managing the trillions of microbes living within. So, the roadmap for developing microbiome therapeutics involves identifying problematic microbial enzymes, designing specific small molecule inhibitors or targeted probiotics, and personalizing prescriptions. Will smart microtherapeutics become part of standard daily pharmacy practice? The evidence shows we are closer than ever. Thank you all. Thank you for watching this video.